Welcome everyone. Today we are going to learn about basic facts on transport. Now the first question that comes in my mind, why transport is important for a cell? And the answer is actually depends on plasma membrane. We know plasma membrane forms permeability barrier. Now the plasma membrane surrounds a cells and separates cytosol from the exterior environment, and it actually defines the cell's physical and chemical boundaries. Now, the plasma membrane actually prevents unassisted movement of most water soluble molecules, and it actually essentially maintains the difference between chemical composition of the cytoplasm and the extracellular fluids. As we know that some molecules actually permeable through the plasma membrane, like gases are permeable, CO2, N2O2 are permeable. Now the plasma membrane, as because they are made up of lipid molecules, so they are hydrophobic in nature. So fat soluble molecules are actually permeable through the plasma membrane, like steroid hormones are permeable. But polar molecules, polar means that are actually water soluble. So water soluble molecules are not permeable, but small uncharged polar molecules like ethanol is permeable. Water and urea are slightly permeable, but large uncharged polar molecules like glucose, fructose are impermeable. All ions are impermeable, and charged molar polar molecules like amino acids, ATP, glucose 6 phosphate, proteins, and nucleic acids are impermeable. As because some molecules are permeable through the plasma membrane like gases, small uncharged polar molecules, steroid hormones, and some molecules are not. That's why plasma membrane is called semi-permeable membrane. So the reason for the transport or the importance of transport relies on, as because plasma membrane actually par, uh, actually prevents some movements of molecules or ions. So here comes the role of transport proteins. So movements of impermeable molecules or ions are carried out by transport proteins, and here the importance comes of the transport. Now there are three main classes of transport proteins present. It is called channels, transporters, and ATP powered pumps. So this is the example of channels and transporter. Under transporter there are uniporter, that means one kind of or one specific types of molecules actually transported through this transporter, so it is called uniporter. But in case of symporter and antiporter, two different molecule or ions are actually transported, in case of symporter in the same directions, or in case of antiporter in opposite directions. It could be inside or outside, whatever. So this is example of channels, transporters, and there are ATP powered pumps. So these are the main classes of three main classes of transport proteins. In case of ATP powered pumps, ATP hydrolysis occur. Now I will make separate videos on all these three different transport proteins in separate classes. Now all membrane transport proteins are integral membrane proteins. In my membrane plasma membrane lectures, I have mentioned that there are three category of proteins present in the plasma membrane. One is called integral membrane proteins, another is lipid anchored proteins, and third is peripheral membrane protein. Now, all membrane transport proteins, that means channels, transporters, whether it is uniporter, symporter, or antiporter, and ATP powered pumps, all proteins are integral proteins, and they contain multiple membrane spanning segments. So it is multipass membrane proteins. In my plasma membrane video, I have mentioned. So this is an example of integral membrane proteins. Now, integral membrane proteins are also called transmembrane protein because they traverse or span the lipid bilayer. That's why it's called transmembrane protein also. And in integral membrane proteins, there are three segments. If this is the exterior of the cell, we call this exoplasmic domain. This is the transmembrane domain, and this is the cell cytosolic domain because it faces the cytosol. Now here, this is the transmembrane domain, or that actually cross the lipid bilayer. As you can see here, multiple membrane spanning segments present, and predominantly this portion of the protein is actually alpha helices, secondary structure of protein, alpha helices. Now, in case of these transporter proteins, conformational changes are essential. Conformation means structure. So during transport, structural changes occur of these proteins. So conformational changes are essential to the function of all transport proteins. Now, transporters can walk in the both directions. Both directions means this transporters can move molecules or ions from inside to the outside or outside to the inside. Now, one thing I want to mention that here transporters means specifically uniporters, because in case of symporter and antiporter, that becomes complex. But in case of uniporters, the uniporters can walk in both directions. That means the molecules can come inside or come outside, and that depends on the chemical concentration gradient of the molecule. Now, X-ray crystallographic analysis revealed that transporter means uniporters, are built from inverted repeats, and it is called pseudosymmetry. How it looks? It looks like this. Let's suppose this is the one example of uniporter. Now, the half portion of the protein, if suppose these portions actually helps the molecule to come inside of a cell, the other half portion of the same molecule or uniporter will have the domain that will help the same molecule to come out, and the direction of the movement or transport of the same molecule or the ions depends on the chemical concentration gradient. That means whether the molecules is higher concentration in outside or inside. If the molecule is higher outside and then the inside, then the molecules come inside of the cell. And the opposite, if the opposite is true, let's suppose the concentration of the molecule is higher inside and lower inside, then the molecule will move from inside to the outside. So one uniporter can move the molecules or or ions in both directions, so they are capable of this. So they are basically inverted repeats contents, and it is called the structure is called pseudosymmetry. Now, another important facts on about the transport we have to understand at first, then I will actually go further more details about the transport, is that there are two kinds of movement occurs. The first is downhill movement. Now, what is downhill movement? As the name suggests, down the hill. That means when someone actually come down from a hill, it actually feels easy, okay? But climbing up a hill is actually requires energy, and it is called uphill movement. So there are two type of movements occur for a molecular ions in case of transport. One is downhill, another is uphill. Now, what is downhill movement? Here molecules or ions transported down their chemical concentration gradient. Down their chemical concentration gradient means from higher to lower concentrations. When molecules or ions move higher to lower concentration, there is no requirement of external energy input. Let's see the examples of downhill movements, like the molecules that can traverse directly from outside to the inside or inside to the outside is called simple diffusions. Here transporter is not required, like molecules like steroid hormones and other hydrophobic molecules or movements of urea. These are actually occurs by simple diffusions, or the gases that actually move through the plasma membrane. So these are simple diffusions. And another is actually mediated by channels. So channel mediated transport is also downhill movements. That means the movement occurs from the molecule occurs from higher to lower concentrations. And transporter mediated, here transporter means uh, uniporter specifically, because in case of symporter and antiporter, the mechanism is actually quite complex. So I will make a separate video on this. This is the example of uniporter, where the molecules actually move from higher to lower concentration. So these are the examples of downhill movement. As you can see, the concentrations of these molecules are actually higher in outside. Let's suppose this is the outside of the cell, and then the inside. So this is the chemical concentration gradient, and here downhill movements occur. This process is thermodynamically favorable, and there is no requirement of external energy, so it's called passive transport. Now, there, as I have mentioned, there is another kind of movement occurs. We call it uphill movement. Here you can actually make an analogy that when anyone climb up the hill, it requires effort, it requires energy. So here the molecules or ions are transported against their chemical concentration gradient. Now, Why the molecules or ions are transported against their chemical concentration gradient? Because it is not necessary that always the molecule should transport from higher to lower concentrations, because sometimes it is essential to move some molecules or ions from lower to higher concentration also. So now this type of movements or transport is called uphill movement. Examples like here, in this case, an external source of energy is required to move the molecular ions from lower to higher concentration. As you can see, this is the um, concentration gradient expressing that these portions, or the, let's suppose this is the cytosolic portions of the cell. So here the concentration of the molecule is low than the outside, but if the cells want to move these molecules or the ions from inside to the outside, then it requires energy, because this process is thermodynamically unfavorable, and it is called active transport. So I think you have understood what is passive transport and active transport, because you can easily understand this by two movement. One is called downhill movement and another is uphill movement. So these are the basic facts on transport. I will make separate videos on other points of the transport, like channels, transporters, and ATP powered pumps. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching, and always keep learning.